Hello viewers and subscribers of Biotecnika. I welcome you to today's video, which is regarding a series that we will be conducting, which is on CSIR NET Live Science important questions from previous year. We'll be discussing along with the solution. Today's video is specifically from Unit 5 and that too questions from tetrapod limb development. The limb development, it comes under subunit C under Unit 5, which is developmental biology subject of CSIR NET syllabus. The students which are the aspirants of this exam who are having any zoology background or you have studied developmental biology before, then definitely you must attempt for this unit because it is very scoring. It has lot to remember actually, many gene names and many proteins, pathway, signaling, everything. But then again the questions are quite predictable, and out of that tetrapod limb development is a very important concept which nowadays they are giving lot of questions from. So just to give you a brief overview, what is limb development? The vertebrate limb is an extremely complex organ with an asymmetrical arrangement of parts. And different questions will come from here. What are the genes involved? But then you will also have questions on the axis specification, how it is done. And you have for any part which is coming out from the body, they have a very important axis called as proximal distal. Proximal means the part which is closer to the body and distal means the part which is away from the body. Okay, so always the any such limb will come from proximal to distal, and that is why the parts will be laid down. Initially proximal parts will be laid down, later the distal parts will be laid down. Then you have anterior posterior, where you have the thumb. If you take your hand, if you take the thumb, then the thumb will point towards the anterior and the pinky finger, it will be the posterior part. And another one which is the dorsal and ventral. So dorsal means the backside of your hand where the knuckles are there, the ones which you use to hit someone, isn't it? So that is the backside of your palm, which is backside of your hand, which is the dorsal side, and the palm where your lines are there, palm lines are there. So that is what is the ventral side. Now these all different axes, they have different morphology, and that is why the limb development is a very complicated process. Okay, but however we have some quite some progress which is made in this research area. How much now we know, that is quite a lot. There was a question in September 2022. In mice, the gene encoding TBX5, it is transcribed in the limb fields of the forelimbs. Forelimbs means the ones which come out from the upper portion of the body, while the genes islet1, TBX4 and PITX1, they are expressed in the presumptive hind limbs. Hind limb means the ones which is in the posterior portion of the body. So these many statements are given. Following statements are made about the limb development in mouse. So what will happen, which out of this will represent a correct combination of the statements, right? Now this is obviously a little bit of common sense also, because it is clearly mentioned the gene encoding TBX5, it is transcribed in the limb fields of the forelimbs, right? So if it is written loss of TBX5 results in complete failure of forelimb formation, this absolutely makes sense because it is a very important gene in making the forelimb, right? So that's why definitely A will be our answer. And apart from that, hind limb bud growth and initial patterning appears normal when TBX4 is knocked out, although leg development is arrested prematurely, right? So we know that TBX4, it is needed in the hind limbs, but it is said that initial patterning, if it is even if it is appearing to be normal, but the leg development majorly it depends on the TBX4. Leg means the hind limb. So initially even if it seems normal because of some other genes, but it will be arrested beforehand itself. Okay, so it will not continue to make, right? So definitely A and B is our combination, but we will also have a look at the other two regarding PITX1 and islet. Now the gene encoding TBX5 is transcribed in limb fields of the forelimbs, while the genes encoding islet1, TBX4 and PITX1, they are expressed in presumptive. So obviously option A is correct, that loss of TBX5 results in complete failure of forelimb formation. And as I already mentioned, this is actually taken out from NCBI paper. So hind limb bud, that is a Gilbert book. This bud growth and initial patterning appears normal when TBX4 is knocked out. Knocked out means it is removed. So the gene functioning won't be there. But the leg development will ultimately be arrested. Okay. And if PTEX1 is misexpressed in the mouse forelimb, it will cause the muscles, bones and tendons to develop, right? When islet1 is inactivated specifically in the lateral plate mesoderm, hind limbs will not form, and this is consistent with the role in hind limb initiation. If you have a look what is the role over here, you can see that the this blue color, it represents TPX5. So where is the expression of TPX5? In the entire region where this 4 limb buds have come out. Limbs come out from the buds, okay? And this areas you can see TBX4 is there, and also PTEX1 will also be there, right? And TBX5 expression leads to the forelimb, and TBX4 it will be giving the hind limbs, right? So that's why even islet1 is there if it is inactivated specifically in lateral plate mesoderm. Lateral plate mesoderm means it will be present over here, right? So the one which is present at the sides of the main stem, then hind limbs will not be forming because islet1 is responsible for it is one of the genes involved in hind limb. Okay, so that means it has a role in the hind limb initiation. So it is written over here that when islet is inactivated, the hind limb still develops. So this is wrong, right? And misexpression, it will cause development of muscle, bones and tendons. But here it is written that it will cause the development, misexpression of PTEX1, it will cease development. That is why even C is wrong. Okay. Now let's move on to the next question, which was from June 2019. During the wing development in the chick, if apical ectodermal ridge, which is called as AER, if it is removed, the limb development will also stop. And on the other hand, placing the leg mesenchyme directly beneath the wing AER. So what I showed you, there was a limb bud which was arising. The distal portion of the limb bud, that means the part which is coming out from the body, this portion is a thickened epithelial layer which is called as the apical ectodermal ridge. Okay, so this apical ectodermal ridge is very important because it gives signal to the underlying mesenchymal cells to extend the limb forward. Okay, and even the mesenchymal cells will also give signal to the AER to keep it in a proliferative form. That means it will maintain the AER. Fine. So the apical ectodermal ridge, if it is removed, the limb development will stop. And on the other hand, placing the leg mesenchyme directly beneath the wing, distal hind limb structures develop. So this may demonstrate that whatever based on the above observation, some statements are given, which combination of the above statement is demonstrated by the experiment, right? So actually this is from an experiment which was already conducted, and it is a very popular diagram in Gilbert's book, all those who have already prepared from it. Here we have the forelimb mesoderm, and then AER is the front portion. So if AER is removed, then it is the pivotal role in extending the limb. If AER is removed, then limb development will also stop. The proximal distal growth and differentiation of limb bud is made possible by a series of interaction between this AER and the limb bud mesenchyme. If you put some extra AER, then you will see that the wing will be duplicated, because at the distal end, as the limb bud is extending, the AER will be present towards the distal side, towards the side which is away. So from there, you will see that at the distal part, the wing is duplicated, right? And if you put leg mesoderm under the AER, then although the limb starts as a forelimb, that is wing, but at the end part, since it will have the leg mesenchyme cells and it does not get signal from the side of the body because it has already moved away. So at that point of time, half of that will develop into leg, right? If you put any non-limb mesoderm, so they don't have the signals to make into limb-like structure. So in that case also AER will slowly start to regress and the limb development will completely stop. Fine. But why AER is maintained? It is because of FGF, which is released from the mesenchyme, fibroblast growth factor. So if you remove the AER as before, but in turn if you put a FGF bead over there, then the epithelial layer can be made into an AER once again and normal wing will be able to form. Okay. So although the mesenchymal cells induce and sustain the AER and determine the type of limb to be formed, but the AER is responsible for the sustained outgrowth and development of the limb. Okay, so AER keeps the mesenchyme cells directly beneath it in a state of mitotic proliferation and prevents them from forming the cartilage. Because if it forms cartilage, it cannot extend any further. That is why AER keeps the mesenchymal cells underneath in a proliferative phase. Okay, so according to that, you'll have to see what the description is telling. Obviously, if apical ectodermal ridge is removed, limb development ceases. That is true. On the other hand, placing leg mesenchyme under the wing AER, that is the hind limb mesenchyme in forelimb AER, then the distal hind limb structure, that means the parts which are further away, they will develop at the end of the wing, which means hind limb structures will develop at the end of the forelimb structure. And if limb mesenchyme is replaced by non-limb mesenchyme beneath AER, then the AER is going to regress. It won't be able to extend. That means what? The limb mesenchymal cells, they will induce and sustain the AER. So this is a correct statement. The mesenchymal cells specify the type. Obviously, whether it will form wing or limb, that is why even B is correct, because if it is not there, then because of that, limb is not able to be made. Okay, so that means mesenchymal cells are the ones which are specifying. Whether AER is there or not, we need the mesenchymal cells to have the signal to form either wing or limb, right? AER is responsible for specifying the type. No, this is not correct, because even if AER is there but mesenchyme is not there, still we saw that neither wing form nor limb form. That is why C will not be right. Okay, so that's why 1 cannot be correct. Anyways, A and B both should be correct. So second will be a right choice of option, right? Then the AER is responsible for sustained growth and development of limb. Yes, because AER will maintain mesenchyme and mesenchyme will maintain AER. So both are reciprocally maintaining each other. And AER does not specify the type. So here you can see C and E are opposing statements. So yes, AER is responsible, AER is not responsible. If AER is not responsible, is wrong, that means AER is not responsible, this will be right. So D and E is also right. Second is the right choice of answer. I hope that was clear. Now let's move to another question that is from June 2021. Now, which of the following statements is not correct regarding the tetrapod limb development? So here we see some of the concepts such as ZPA, then SHH, then something regarding WNT7A. It is saying as the limb grows outward, the stylopod forms first, the zygopod and the autopod is formed last. Each phase is characterized by a specific pattern of Hox gene expression. So definitely this is so you have to mark not correct. Okay, so this is a correct statement. Why it is correct, you just have a look over here. I showed you the diagram in the first slide. There we had the first part of the limb, it is called as a stylopod. Okay, and then that is humerus, what we have in our forelimb. Then we have zygopod, which is the radius and ulna. Okay, and then we have the autopod, which contains the carpals, that is the fingers. So as the limb grows outward, first obviously the proximal parts will be laid, as I mentioned. That's why the stylopod will be forming first, then the zygopod will form, and then the autopod will be forming last. Each of these phases involves the expression of Hox genes. Okay, so that's why the first statement is correct. Then we have the zone of polarizing region. So what is ZPA? Whenever you have this limb bud, okay, now limb bud will have an anterior axis formation and posterior axis formation, right? So this at the posterior side, you will have the expression of something called as ZPA, because of which it will give the AER a signal so that this area will form the posterior parts, okay, and this area will form the anterior part. So how it is maintained? It is maintained by the interaction of fibroblast growth factor from the AER and SHH, which is expressed from the mesenchyme. So the mesenchymal cells which are present near this area, they are going to express SHH, which is going to signal AER, and AER is going to give further signals forward. So even second is the right option, means correct statement. The ZPA is established and maintained by the interaction of FGF8 from the AER, and the mesenchyme made competent to express sonic hedgehog by its correct expression of particular Hox genes, right? So you can see here, this is the AER of the limb bud, and this is the progress zone. That means it contains proliferative mesenchyme. So ZPA gives this ZPA is present in the posterior area, not in the anterior area. Okay, and if you see here, these are the interactions with the ZPA and AER. So ZPA releases SHH, as I said. ZPA will release SHH and it gives signal to the AER. And what is AER containing? Because of that, what it is expressing? FGF4 and FGF8. Because of that FGF4 and FGF8, it is able to maintain the ZPA and continue the expression of SHH. So why do we require continuation and maintenance? We saw that in case of mesenchymal cell also. We are seeing this in SHH also, because limb bud is extending further and further. It's not one time signaling that is done. Okay, so as it is extending, even here also posteriorization needs to be done. Here also posteriorization needs to be done. And as I showed you, at the end of the limb also, your pinky finger will be the posterior, whereas the anterior will develop thumb. So who will give these signals? That is why a continuing maintenance will be required. Okay. Then you have the dorsal ventral axis formation. So as I told you, you have different types of axis formation questions. Here the dorsal ventral axis is formed in part by the expression of WNT7A in the dorsal portion of the limb ectoderm. So how is the dorsalization done? By something called as WNT7A signaling on the ectoderm. Now because of this WNT7A, it will maintain the expression of SHH in the ZPA and FGF4 in the posterior AER. So this is what they are saying in the fourth statement. That's why it's correct. Dorsal ventral axis is formed in part by the expression of WNT7 in the dorsal portion of limb ectoderm, and that will maintain the expression level of SHH in the ZP and FGF4 in the posterior AER. Okay. Then another thing about this is regarding cell death. So this is regarding the digit formation. So as you can see, almost entire limb formation is asked in this question. Okay, all components. Cell death in the limb is necessary for the formation of digits and joints. It is mediated by bone morphogenetic proteins. The effects of BMPs can be regulated by Noggin protein, and the BMPs can be involved both in inducing apoptosis and also in differentiating the mesenchymal cells into cartilage. Okay, so it as we saw that it is mediated by BMPs, but in option third it is saying although cell death in the limb is necessary for the formation of digit and joints, it is never mediated by BMP. No, cell death is necessary, but it is very much mediated by BMP. Okay, so it is also responsible for differentiating mesenchymal cells. So it is not only responsible, it is also, along with that, it will also cause this cell death in the limb. So that's why 3 is the wrong statement. So as you can see, regarding axis development, lots of questions can be asked. Then also in June 2021, there was another question on limb development. Okay, so you have to match the different parts of the limb area, whatever things are there, with what is the description over here. Different parts of the vertebrate limb development with their description. So this is quite easier, I would say, because if you know 1 or 2, you will be able to figure out the rest of the part. So here we can start with this mesenchyme, which we already know. So what is mesenchyme? It is loosely organized, mainly mesodermal embryonic tissue, which is giving rise to the most of the parts in the limb. So B will be 3, and that's why you have this first option, which is correct. Okay. Now we can see the others also. We know what is AER. Now AER, we know it is the thickening of the ectoderm. Ectoderm means the outer layer of cells. So this is the thickening at the apex. Apex means the tip of the developing limb. So these two anyways you would know if you have studied a little bit of limb development. So anyways, knowing one also you can figure out the answer. So C is 2 here and C is 2 here also, but B is 4 here. That's why this won't be the answer. Okay. Rest anyways, let's explain. EMT, it's epithelial to mesenchymal transition. See, some cells will be in layers. So those cells, they are called as epithelial cells. Okay. Whereas some cells will come out of the epithelial cell as loose cells. So those are called as the mesenchymal cells, right? So it will come to here. Epithelial cells making up the mesoderm of the early somatopleure undergo the transition and get included in mesenchymal cell pool. That means they come out of the layer and they become loose cells. Okay. Now what is progress zone? Progress zone is the proliferative mesenchyme. It is a mesenchyme only that fuels limb bud growth. It is just present just below the AER. What is ZPA? Just now we saw it is required for the posteriorization. So it's the cells found within the most posterior region of the limb bud. And autopod, it is the distal part of the tetrapod limb, the most end part where the fingers are formed, right? So these are the different explanations as we just saw. Let me know in the comment section if this video was helpful to you, and also which topic under Unit 5 you want me to discuss the questions from. The previous year questions, whatever questions important have come from that particular topic, I will discuss on this channel. If you put in the comment section which topic you want to know. Okay, thank you everyone.